I'll just mention first, oh, got it, recording in progress. There is a bioRxiv preprint, which I know Ludwig has sent around the link to. So if you want to know more about this or see in more detail while I'm giving your talk, you can go ahead. Ludwig has been involved in this as well, and other major participants as well are Sean Davis and a few other people in my lab, and these are some people from the Galaxy project who have contributed to a Galaxy component for this. So just to give an overview of the purpose, we all know there are a lot of gene expression profiles deposited in public archives, but many of them remain unused when it comes time to interpret new experiments. And the motivation here is to leverage these databases in comparative analysis and allow us to interpret new experiments by comparison to those that have been done already without a lot of effort by the user, heavy computational resources, or being restricted to specific contexts. So the goals here are to first provide a model that is pre-trained on large heterogeneous public data sets and our Bioconductor package, making it easy to apply this interpretation model on a new data set. And I'll get into detail about what that means shortly. And through all of this, enabling users to interpret a new data set in the context of these of a variety of existing databases. So I'll put this in some context of previous studies, talk about how we build this model, how you can use it, and some benchmarking relative to the most similar tools that are available. So previous studies are a little bit difficult to identify, in that these are not exactly the same thing, but there are two things that are in some ways comparable to what we're doing this, and I'll talk about both of these in a bit. The first one was sort of the methodological, what's the word I'm looking for, inspiration for what we've done here. And the second one, multiplier project from Casey Green's lab, is an alternative approach to matrix decomposition for this approach, or for this objective. So briefly, this CRC subtype project, this was a project that I started towards the end of my time as a postdoc at Harvard, and I passed off to another PhD student, Siwan Ma in Curtis Huttenhauer's lab, who eventually finished it and published it in Genome Biology. And this came out of a study of a meta-analysis of colorectal cancer microarray data sets with an attempt at identifying subtypes, which ended up being instead a proposal that there are no true subtypes of, at least as far as the whole transcriptome goes, of colorectal cancer, and that there are instead some axes of variation that define a range of variation in the transcriptome. And so this was a method that my supervisors and I came up with to learn these axes of variation that were identified through principal components and connect them across different data sets. And the issues that we had here is that there are batch effects. Some data sets were heterogeneous. They might contain things like normal samples, and although those could be removed, there were other sources of heterogeneity in the data sets as well that were hard to know where it was appropriate to filter these data sets. And so this approach involved, it was a fairly bespoke approach with a lot of work on first doing principal components analyses, taking the loadings vectors, those are the vectors that associate the original gene expression values to the principal component latent variables, treating those as observations or just as vectors that were then clustered across studies to identify clusters of these loading vectors that were present in multiple studies. And this turned out to have some advantages that we studied in detail in that manuscript. For example, things like irrelevant samples in a study, normal specimens, or large batch effects would essentially be ignored and would have the effect of potentially changing the order of where a principal component occurred in a data set, while not polluting it too much and not losing it. And this extended even to extreme cases of different microarray technologies, use of formalin-fixed samples, and the inclusion of RNA-seq as well. So we got the idea here that this was a pretty robust approach to learning axes of variation that existed across multiple studies. So that is just a bit about the background of this. And then Plier, this method that I mentioned, which has been done to enable transfer learning from data sets. And when I say transfer learning, I just mean in general the transfer of knowledge from one context into another context. This is just another matrix decomposition, but where a set of pre-specified pathways or gene sets are used to constrain the dimension reduction onto axes that are defined only by genes in those gene sets. So it's a more sparse dimension reduction than principal components analysis, but accomplishes something similar with ending up with a pathways by samples approach. And then there was a follow-up to that original Plier paper called Multiplier, where they used a large collection of uniformly processed data to do something similar and get these they called them LVs, latent variables, that would allow you to project a new transcriptome data from a new data set onto these interpretable latent variables that had been defined in this large transcriptome data set. They used Recount2, which is a big curated RNA-seq data set that we have also taken advantage of. So now genomic supersignature. It is basically as I briefly described before, involving a principal components analysis in each data set, taking the rotation vectors from each of these principal components analysis and from each study, and then doing a clustering of those to get what we finally settled on calling RAVs, for replicable axes of variation. So they're replicable because they are identified in at least 2 studies, either 2 studies in the database or 1 study in the database and the study of interest. For the user, when it comes to applying this, they bring a data set of study, have these, this is just a simple principal component analysis, which is implemented in the package, and along with these data sets, which are this pre-trained model or RAV model that we're calling it, which involves all of those procedures that I mentioned on the previous page of principal components analysis, and then clustering on a large on a large compendium of RNA-seq experiments to get this output where each of, well, there are several outputs that I'll show you later, but the most simple one would be taking the samples of your studies and getting a new representation of your data in terms of these RAV scores that have been predefined from this compendium. And this could be, so these are not necessarily being calculated on your data set, that you're either matching your principal components against ones that have been observed or calculating ones that have been observed based on those rotation vectors in your data set. The second, so part of it is this this approach of dimension reduction and creating this index from publicly available data. The other part is efforts on annotation, and this is where, as Seun has, there are a couple of ways that she's taken this to the next step, both beyond what we had done back on CRC, and in addition to applying this to a much larger data set and to what these other efforts from Plyer have done. So one of them is the methods for annotation. So these RAVs are, as part of the model building process, they're pre-annotated from a couple of sources. One of them is PubMed mesh terms. So every RAV comes from certain studies. So we know which data sets and which principal components contributed to that RAV. They were part of the cluster that created it. And every one of those studies comes from PubMed and has these medical subject headings. So those are pre-existing annotations that are in this pre-built model. Another one is through gene set enrichment analysis. So these loadings provide what you need to do a pre-ranked gene set enrichment analysis, and all of those are pre-calculated. So based on the order of the magnitude of loadings, they each of these axes is annotated with gene sets. There are a couple of options that can additionally help you to interpret what that axis means. And then these components here, these are applications. The metadata there, each of the RAVs have searchable metadata from the mesh terms, from the titles of the PubMed entries. So you can search those for things like, this is an example I'll show later, neutrophils, and find axes that are known from their metadata or can be suspected to be related to some metadata attribute of interest. And then this finally is a matching of the principal components of a given data set to the most closely related RAVs from the database. I guess I won't go into detail about this, but the model building process starts with this large RNA-seq compendium, goes through some filtering, transformation steps. I should mention that all of these things Seun has done sensitivity analyses and found the process to be quite robust to all of these selections, such as transformation and normalization. So these are for each study, principal components analysis performed, and then clustered and built into this model. So our source data right now are this RNA-seq sample compendium from refine.bio, about 45,000 samples. We've tried things between the top 8 and top 20 PCs. It doesn't matter too much what the cutoff is here, because the very high PCs tend to be selected less or replicated less across studies. The hierarchical clustering produces on the order of 5,000 clusters, including a number of singletons. And within each of these clusters, the principal components are averaged, and that's what produces your RAV. So it's just an average of some closely related principal component loadings. The mesh terms are just annotated based on so they're looked up for each study that belonged to a RAV, and then counted and annotated, weighted by the frequency of the term for this RAV, and the variance explained by the PC for which it appears. So something that is a very small component of the data set will have that mesh term will have less weight than the first one. Again, so the gene set analysis, all of these are included in the models, along with enrichment scores. Since this is a bioconductor group, I'll just mention a little bit about the package itself. The PCA genomic signatures, it's just a lightweight inherited object from summarized experiment that it doesn't change the class. It just mostly defines a number of functions that can be added on it, so that this summarized experiment contains both all of the RAVs, which we call the RAV index in the assay slot. It contains information on each of those RAVs, such as GSCA results and the studies from PubMed associated with each of those RAVs, along with metadata about the model itself and about the clustering that went into it. So this is in Bioconductor now. I'll just talk a little bit about how to use it. So here is an example of a data set with some isolated immune subsets, including CD16 neutrophils from patients with autoimmune disease. And this is all of the examples that we've done here, we've taken from the publications of these previous works that I told you about. So they're not cherry-picked examples for this tool. They're things that have been that were used for demonstrating the efficacy of previous tools, and we've just reused them to show how this compares to those previous studies that I showed you. So this demonstrates just a few of the major ways that you can use genomic supersignature, starting with the matching to existing existing RAVs. All this is, data set, is your data set of interest. This is a summarized experiment using by default gene symbols for gene names, and a RAV model, which can be, there's an example one given in the package, or it can be instantiated with just one command. It's about, I think, 0.5 gigabyte, the full RAV model. This can potentially be multiple data sets here, but what this heat map shows for each of the data sets, just a, in this case, it's showing the silhouette width and the scores. So the score is the correlation for the loadings of a principal component in this data set to one of the RAVs in the pre-computed model. And the silhouette widths are showing you something about the coherence of the RAV from that data set. So a high one like this shows this is a fairly compact, coherent cluster. Some of them are less compact, less coherent from the literature. This word cloud, so it's made here for one of the RAVs with the top score, the top correlation to a principal component in this data set, and these are based on the mesh terms from the studies that went into this RAV 1551, and we see here a number of blood-related terms. So these are things that if you were to go and look at the PubMed pages for each of these studies, you would see pretty clear that these are topics represented in the title and abstract of those studies. And then this plot of the validation study is gathering a whole bunch of different things together. The cluster size, this is the number of principal components from the literature that contributed to a RAV. The principal component from our study that's being matched, and we're seeing here the silhouette width and validation score. So these ones up here, these are very closely related. Validation score of above, what is this, above 0.7. So that's a correlation of more than 0.7 to a principal component in our data set. And this large size shows that this is something that came from somewhere about 15 principal components from the published data sets. And then once you have identified a principal component of interest in your data set, so this is PC2 from our data set, it's allowing us to annotate this based on the components that it has matched to in the data set. So this is something fundamentally different than if you were to how can I say this? If you were to just do this based on your own data set and your own analysis of the principal component loadings, it would not be based on some axis that has been established based on a number of previously published data sets. I mentioned that validation function could take multiple data sets. This is an example here where we took 5 different data sets from TCGA, and ran each of these. So this is a list of these 5 expression data sets and matched it against RAV model in order to try to identify components that are maybe not specific, that are present in all of them, and ones that are specific. So we find here, and this is just the top listing of RAVs based on ones with the top scores. We see clearly there is one RAV that is fairly specific to breast cancer, and one that is fairly specific to colon adenomas and adenocarcinomas and rectal adenocarcinomas. And if we look those up, we do see that those principal components from the literature are associated with publications that do have to do with breast cancer. In the case of the one that matched most closely to breast cancer and to colorectal cancers in the other case. So this is the word cloud. These are the actual studies here with titles and enriched pathways. So a little bit of benchmarking against these other studies that I mentioned. So the first one is kind of interesting to benchmark against, I think, because for a couple of reasons. One, it was a bespoke analysis of 8 colorectal cancer data sets, so it was really focused on this disease, and it came from microarray. So this is, completely different technology, completely different purpose, aimed at defining axes of variation in colorectal cancer. And then the second one is also based on a large RNA-seq compendium, but using this fairly different method and our approach of PCA and a hierarchical clustering that we ended up settling on. So here are a couple of comparisons to these previous studies. This first one, all of these are showing the scores. So in this case, PCSS are the ones that were defined by that colorectal cancer study, and these are in a validation data set that was used in this. It was called the consensus CMS, trying to remember. Consensus subtypes for colorectal cancer. This was a pair of Nature Methods papers that ended up defining essentially the same 4 transcriptome subtypes of colorectal cancer that in this PCSS principal component subtype score paper we compared to, and showed that that each of, if we look at these samples and our continuous scores on them and compare these to the discrete scores proposed by this consortium, as well as the ones that they decided not to label as having a distinct subtype, we see essentially a very close association with those discrete subtypes as being in the 4 quadrants defined by these 2 different scores. So just to say that with that study and with ours, with these 2 RAV axes that we identified through the validate method as being similar to those PCSS 1 and 2 scores, both of these are closely related to the discrete subtypes. But are they, so they're related, are they better or are they worse? This was an attempt to assess whether they were better or whether they were more or less representative of colorectal cancer biology by their ability to differentiate clinical features. So this is a likelihood ratio test. The setup here is we have 4 different pieces of clinical and pathological data, and we set up a model where one of these is the outcome variable, and either one or both of these alternative transcriptome models is the predictor. So the case where both is the predictor, this is what I call the full model. And then there's a second, which is just one of these as the model. In the likelihood ratio test, if we look, say, at CMS, it compares. So this would be a model where we predict microsatellite instability status just from the CMS subtypes. These were the discrete ones, and compare that to the likelihood ratio test, where we use both the CMS and these previously proposed continuous scores to predict MSI status. And a small p-value here, so this case essentially means that the full model, adding the subtype scores, is a much better representation, has a very large increase in likelihood of the model compared to using the consensus molecular subtypes alone. And these p-values close to 1 indicates that adding the discrete subtypes to the continuous scores has very little effect on the likelihood. And so this is a case in all of these, those subtype scores based on the colorectal cancer microarray data sets are better representative of microsatellite instability, grade, stage, and tumor location. So we went ahead and did the same thing with these two RAVs. And I think this is pretty remarkable because this is a validation on microarray data sets. Not a single microarray was used in the training of this model. And also, this is trained on a very large unfocused database. So there are some colorectal cancer studies in it, but it's in no way focused on colorectal cancer. And essentially, we see nearly the same thing. These RAVs perform almost as well as the focused PCSS scores, and definitely better than the discrete subtypes that were created by a consortium focusing on this project specifically. This second example, the neutrophil-associated signature, is an example that was taken from the multiplier paper in a study of systemic lupus erythematosus. And so there's a data set of this disease, which we mapped to RAVs in our model, and went through these same steps, the keyword search in the enriched pathways and correlation with the data set metadata to identify RAVs that are related to neutrophil count, as was done in this original paper. I added a little example here just to show. So their objective here in this particular thing was finding a way to use transcriptome data to predict neutrophil count in data sets where neutrophils had not been counted, and comparing this both to actual neutrophil counts and also to a model developed specifically for predicting neutrophils, this method called MCP counter. And in the multiplier study, they showed that it performed well both against actual neutrophil counts, against these estimates based on a specific tool, MCP counter, in two different data sets. So we did the same things and essentially found the same results as this study. I think this is only interesting in as much as showing that that using a different RNA-seq compendium, this works as well as was shown in this other study. And we've again rediscovered this transcriptome model that is pretty good at predicting neutrophil counts and doesn't require a neutrophil-specific training data set in order to do it. So that's an example of the transfer learning application. Just a note about pathway separation. So we've done just some summary analysis of these to see what their coverage. They cover about 30% of the MSigDB C2 pathways, and the separation. These were some examples again taken from the multiplier study, I believe. These are just some handpicked, very similar pathways that were looked up and seen that these are separated in the model, meaning we have are in C2 type 1 versus type 2 interferon. They're similar pathways, but they're annotated to different RAVs in our model. The same for neutrophil monocyte macrophage related gene sets. They're annotated to different RAVs and G1 versus G2 phases of the cell cycle. So just to summarize, genomic supersignature helps to connect new data sets to existing data through a dimension-reduced index. That dimension-reduced index allows you to do all of this in fractions of a second. So I didn't show any demos of those things, but the longest part here is the download of that index, which on a reasonable internet connection just takes a few seconds, and then the lookups against it are all very, very fast. There is also a Galaxy toolshed model. This I was kind of excited about. We reached out to some people from the Galaxy project who were excited to implement this in Galaxy and tie it in with some of the popular RNA-seq workflows there. I think it compares well to these existing tools. The closest thing we can compare it to is Multiplier. And some of the reasons I think it compares favorably, it provides a flexibility with annotation because annotation is separated from model building, whereas those gene sets are built into the model building process. With Multiplier, it's a post hoc process, and you can really add any source of annotations that you want to this. The only built-in part is this pre-built model of RAVs. You can then annotate those studies or those pre-ranked gene lists in any way you want. It's implemented as a Bioconductor and Galaxy package, and it's fast to use. It's modular and it's scalable. Currently takes something like 2 days for the full model building. That can probably be improved some, but it's we've seen that it scales approximately with the size of the data sets, and I think we could already increase this to something like 20-fold without any impact on the end user. It would require a fair bit of memory to build, and that's it. The package, the source is on Seyoon's GitHub page, and all of the use cases presented here and in this paper are done reproducibility on this nice genomic supersignature paper repo. The future work, this has been on our minds lately because we're submitting a small grant to extend this to microbiome work, and I know some other things that Seyoon is interested in doing is extending it to training specifically on a single cell compendium or on mouse data sets. we noticed that there are some single cell data sets in refined bio, and what seems to happen currently is that they just cluster together into one RAV. They're sufficiently different, and there isn't enough of them in the compendium to get a lot of resolution among the single cell data sets. These are going to be maintained separately, the toolbox as the software, and the databases which live in cloud buckets and are downloaded by the software. And another interesting direction, I think, is ways of manually curating these RAVs. As we study them in more data sets and compare them to the metadata of different data sets, we can learn more about what they are, also by comparison to curated metadata from the existing data sets. That's all I had today. I think I'm right at about the 45-minute mark.
Yes.
I'd be happy to take any questions and discuss, and maybe bring Sayun forward too to help me with any questions.
Great. Thanks a lot for a fantastic presentation, Levi. Yeah, as Levi said, if you have questions for Levi, really feel free to post in the chat or just speak up here. Maybe I start with like something that also Federico had pointed out in the chat. One challenge here seems to be a little bit to get a good intuition what these RAVs, what these replicable axes of variations really are. Federico asks in this context, so how does such a RAV compare maybe to something like a classical pathway?
So a RAV is a principal component in a published data set. So it's not by itself, there's no pathways involved. It's just those loadings would tell you something about a set of genes or whatever observations that were correlated in a data set, and that together accounted for a significant amount of the total variance of gene expression in that data set. So that's all the RAV is. Like the singletons that are in the database, they are just the loadings of from the principal components analysis of a published data set. That's all they are. And then all of the other stuff, the annotation with pathways, annotation with mesh terms, those are just ways to help you try to understand what that RAV is, and things that you could do. While the mesh term thing you couldn't do on your own, but you could do a pre-ranked GSCA on a principal component of your own data set in the same way. Does that help to understand what they are?
I think the question was coming before you actually kind of clarify that it's just a PCs basically.
Yeah.
So I thought of that more like a signature per se, but it's not a signature as it was. So as we as we or the people that we work with actually call signatures, right? It's it's the underlying code for the signature that it's there. But yeah, I can come back later and leave the space for Nico's and Robert's questions. But yeah, thanks, Levi.
You're welcome.
Yeah. So just to recap, so the RAV seems to be, Levi, if I understand correctly, a principal component that we just see over and over again, right? In existing data sets. That's why they are called replicable. And Nico was the next one who had a question. Would you just like to speak up, Nico?
Yeah, sure. Can you hear me now?
Yep.
So if these, you have these like RAVs, and presumably you could assess their presence, kind of like how much you have this linear combination, weighted combination of genetic features of a sample, even if it's not like one of the primordial sources of variation of a sample. So my question is, if you have like a series of RAVs that are related to features that are known to be clinical indicators of something, can you assess multiple samples and like what does the distribution of the RAV scores of different samples for one such feature would look like? Could you say like, if your score in this RAV is above this, you're likely to have like propensity to leukemia, who knows, something of that sort.
Right. So the first part, yes, that's already there. You can take something like that neutrophil RAV or all of the RAVs and just calculate them in your data set so that you have a RAVs by samples matrix. So if you want to estimate neutrophil count in your data set, you just use that same RAV and it's done even on a single sample level. So there's no principal components analysis in your data set. That second one is actually interesting, and I don't think we've really thought about that, like how to say like above this value it's meaningful. Like, you know, if you calculated the neutrophil you know, the neutrophil RAV on hair, you know, you would still get something, but it doesn't mean that there are neutrophils there. And I have to think about that. I'm not quite sure offhand how to say like what a reasonable threshold is to say that this is meaningful. To say that this is actually present, potentially turn it into like a yes, no kind of
the test
thing.
hypothesis.
Yeah.
I just wanted to get your like feeling also
Yeah.
like what the distribution of these look like across samples, like as a monovariate thing, if you take each of them, or how they co-vary, who knows?
Yeah. I mean, we have their distribution, or we can calculate their distribution in the training data set. So actually, if we were to look at their distribution in the training data set, in the data sets that contributed to that RAV against all the others that did not contribute to that RAV, that actually would give a pretty good idea of a threshold. You would see two distributions, and you could kind of put a threshold in between, or fit a mixture model and give a probability or something like this to do that. We have to talk about this more soon, but I think it's a nice idea. And something that we actually could address reasonably easy by looking at the training data sets and the data sets that are part of the RAV and that are not part of the RAV.
Yeah, it sounds pretty interesting. Yeah, let's talk about it a little bit later. Thank you for good feedback.
Thanks, Nico. Robert, would you like to go next? You are muted.
Yeah, great talk, Levi. First, sort of a clarifying question. So the labels that you're assigning to these, they're coming from the experiment. Is that correct? Not from the sort of loadings on the principal components?
There's both. There's the mesh terms that come from the study that they came from, and then there's the GSCA that come from the loadings. Okay.
So one of the problems with principal components, right, is it sort of captures all kinds of underlying variability, some of which, you know, is related to the experimental design, right? Some of it's related to demographics. And so, you know, things like the breast cancer one, it's hard to understand how you are sure that you didn't just sort of capture something to do with being female, for example.
Well, those studies would have been all females, so it's not likely to be some. I mean, it could be variation that exists in females compared to females that doesn't exist in males compared to males, but it couldn't be variation in females compared to males.
Well, but I mean, if if you had other data sets that had both sexes in them, right, then components of variation for that would be due to changes in expression across sex. So now if you take something that doesn't have that, you're missing that component. So you're matching, right? You sort of have a matching problem on both sides of it, I think. And that's not the main, so your big problem with single cell, of course, is, you know, if depending on how you process it, right, Rafa and Wolfgang and others have shown that your sort of principal component number one is typically, you know, the sort of strongly correlated with the proportion of zeros in the data. And so that's, you're going to basically find that things line up, right? Because that's related to the underlying level of expression, right? So you sort of, it just seems that it might be useful to try to think more about what these what might be driving some of these principal components, right? You have the experiments, you know what the demographics are. It might be useful not just to produce principal components, but also to think about whether those principal components are driven by components of the experiment at hand, right? It doesn't have to be totally naive.
What do you mean by the experiment at hand? Like,
well, you know, sex
driven?
and age, and right, and so you just ask the question of, is my principal component related to any of these other features, right, of the experiment, so that you know, if you have one that's driven by age or driven by something to do with experimental conditions, and somebody else matches on that, well, you know, right, they've potentially just found that demographic feature again, not something that's intrinsic.
Yeah, so there's, I guess, two points at which you can do that. We could do that with the training data sets, and as much as we have metadata for them, by comparing these RAVs to the metadata in the training data sets, and then as part of the application to the new data set as well. And I think, you know, Seyoun has done that in some of the, as a manual process for some of what seem like the most important RAVs, the ones that appear, that are in that are PC 1 or 2 in more than a dozen studies. But it's been just a manual process, not something we've been able to do systematically against all of those training data sets. Do you want to add anything to that, Seyoun?
Not really. I don't kind of, yeah, it's an interesting point. It's like different from like a batch effect, that like some factors PC itself is like influence, like kind of confounding effect, like that it might be kind of buried in the process. I cannot really think of how to incorporate that right way to the model, but at least like a little bit of manual creation and have a sense of the level of that, like what is the influence, or like how much, what is the resolution our method has, just to have the, even like if there's a shortcoming, even have the idea of like what it is and how much it is. That can be important. And then I think that part, I think we can do with little additional manual creation. But yeah, I need to think a little more about how to incorporate the metadata into the sample level of metadata into the interpretation of RAV, I think. So that's what I understand now.
Thanks.
Yeah, excellent. So we are actually a little bit out of time, but if we want to take 1 or 2 more questions, Levi, it's mainly your call.
Fine with me.
Yeah. So I think I see there is 1 more question from Federico, but I'm also really interested in asking a question, given that we see here this slide of extending this to microbiome and single cell data. And Robert had already a little bit started on the single cell work. I think you thought already a little bit about the microbiome extension, given that there's already a grant. So I think it's yeah, not straightforward, but I would see some path on both of these approaches. I think for microbiome, what's your database? How do you exchange like Refine Bio? I think you just take curated metagenomic data, right? And then I think like when I look at the transformation and PCA, so people do PCA for microbiome data a lot, right? But they kind of like currently there is this centered log ratio transformation that people seem to apply. So you would maybe exchange that instead of the shifted logarithm that Seon applies, right? And
there are a few options.
Yeah.
Yeah, there are also some sort of microbiome specific alternatives to PCA that as long as it provides loadings, that's what is important. We want to be able to relate the RAVs to components in individual data sets, and to have in the case of microbiome species and genus labels as variables to allow you to cluster them, calculate them in other data sets and interpret them. But yeah, there are options.
right.
Say you can try a ton of different things for RNA-seq, and it will require something similar for microbiome as well. Just saying, okay, how does the clustering work? Are the clusters, are they all singletons? Are we actually getting some clusters that come from different studies? Are they coherent? Do they seem to have some interpretability? Things like this.
Mm-hmm. Right. And yeah, so similar, like I'm thinking also about the challenges when then apply that not for bulk RNA-seq, but single cell RNA-seq. And there again, there is now a little bit of movement away from these UMAPs, but rather to like Rafa, as Robert said, did this GLM PCA as a generalized version of PCA that could be maybe used here for single cell data, or Wolfgang Huber, together with one of his PhD students, Konstantin, I think, just put out this paper, which basically saying if you do your shifted logarithm and standard PCA, you are actually in a pretty good spot. So that seems to be pretty much what Seon did here. And then the question is which database you maybe use, what's a good selection for a public single cell database. I don't know. The things that come into my mind is something like Pangloa DB or the EMBL single cell expression atlas. So yeah, so I was just wondering about what are the challenges there going from bulk RNA-seq to microbiome and single cell. And for both, I see kind of like a path to go there, mainly along what kind of transformation do we do and what kind of resource do we need, and do they provide the data sets processed or already as RefineBio does, right?
Yeah, the good news there is when it comes to the technical parts, at least in RNA-seq, like what we saw was very good insensitivity to those methods. I mean, whether we put the RNA-seq through a variant stabilizing transformation from lemma or some packages that do this, or just did a log2 of counts +1, they produced very similar results. So in that case anyways, the results were pretty robust to those decisions, and I hope that they will be in these other instances as well, that as long as you choose something reasonable, and you do see things that are replicated across studies, that you will be able to see them again in other studies, and you'll be able to interpret them. And then from that, the biggest challenges are other than that, finding the data sets that are large and heterogeneous enough, and figuring out how to interpret them, because that's where, like, it's going to be very different in every circumstance. We've proposed in the extension to microbiome to again do GSEA, but using BugsigDB, of course, and
very nice.
Yeah, other sources. But,
Great. Thank you. Yeah. And we have a last question, I think, for today from Federico.
Hi, Levi. I'm trying to figure out one way how, say, for example, the final end user, so the ones that actually have to do the interpretation for this data, can use this in a for them usable way. And I think that this might be probably the output of the GSEA function called on the RAVs, right?
If you're asking like, how does the end user apply this, apply genomic supersignature, or?
Yes, more like apply that, but in a way that you say that you get to biological findings, let's say, because the application is probably well described in the vignette, as most of the Bioconductor packages. But in this case, it's more like, how do I get let's say, a table of some signatures that I can try and interpret, right? And that's probably the output of this GSEA call.
The GSCA that I showed is already pre-computed into that model. So when you download the model, all of that is pre-computed. You don't have to do GSCA yourself.
Okay, so you're not doing it at all from the scratch. It's more like a.
It's just a summarized experiment with genes and rows. That's all.
Okay. Perfect. So I mean, there I don't remember in the slides that you had. Did you had, for example, the genes from the leading edge in that as well? Because if you did, there could be some kind of one step to put that into GenTonic. So speaking in a very biased way in here, but if you had this information, then you don't need to recompute anything, and you can just say, oh, it's just like my DE genes, or like the relevant genes. And if you do have those, then that's, let's say, there's a whole new world out there to explore that interactively as well.
I'm not sure if we get leading edge genes from a pre-ranked GSCA.
I mean, you could do, right?
You could.
At least.
Yeah, you could. I mean, I think Sion called something like FGSCA here, or indirectly through cluster profiler, Sion, if I'm correct, and you could get the leading edge out of there.
Okay. Yeah, it's cluster profiler, and then okay. Yeah, that's the part that I'm thinking on my to-do list, like expand the GSCA, like more, like adding more visualization and then more interpretable output. So right now it's just have all pre-computed GSCA result is part of model, just like a table. So as far as you find the web you wanna like know more about, like GSA is there, but it's it's still a little bit just table format right now. So I think incorporate those like leading edge genes and other things. Yeah,
I mean, in the end, in the normally way one would get that back from FGSCA would be let's say structured list. Let's say it's a mini list separate. It's not a list, but it's more like separated by comma or with a slash, and then you can process that. And if you have that, then let's say I would be more than happy to see that. And that's probably okay. It's probably just the matter of rebuilding that and putting that into the final object, which might also increase the size of it.
Oh yeah. Okay, got it. Okay. Yeah.
But let's say it would give people some kind of immediate way of saying, oh, these are the genes that I'm actually focused on to, and then we can continue that offline. I think we're mostly on the biases.
Yeah, that would be really good. Like, yeah, I myself is not using GSA that much for my own research, so I don't know which one will be the information that people want to like access directly. I mean, so like we are providing all those things like pre-computed, and then I selected something just most basic part and incorporate that into the model. But yeah, if there is like more feedback on like which one will be more helpful, that will be great. And I can, and then as Levi said, it's modular, so I can just update the new version of RAB model that has like, oh, this element is like added, something like that. So that's should be fairly straightforward.
Okay,
good.
Yeah, sure, cool.
And
happy to follow up.
If you wanted to do it today, I mean, all of those loadings are included in this summarized experiment derivative that you download. So you could just take that ranked list and use whatever gene sets you wanted and whatever method you wanted.
FGSEA right now, and it would work, right? So it's more like, yeah, it was just for the commodity of having it pre-computed, which would be, let's say, the beauty. Wow, cool.
Yeah.
Excellent. Then thank you very much, Levi, again for the talk. Thanks everyone for the discussion. And I hope to see everybody soon on Slack or by other upcoming seminars or individual discussions. Have a good rest of your week, everyone, and see you soon.
Thanks, everyone.
Thank you. Bye. Bye. Thanks you.
Bye.